Everyone, welcome to biology class. In this class, we are going to see chapter 5 plant tissue culture, 12th standard botany. So, what is tissue culture? Growing plant protoplast. Protoplast is nothing but the plant cell without a cell wall. It is called protoplast. So, growing the plant protoplast, cell, tissue or organ away from their natural or normal environment. That is, under the artificial condition, if we grow, then it is called as tissue culture. And it can be also called as in vitro. In vitro, it is a Latin word. It means in glass or in test tube, the growth of plant protoplast, cells, tissues and organs. So if we are doing it in a laboratory under artificial means in a controlled condition, then it is called as tissue culture. So with respect to plant, a single explant can be multiplied into several thousand plants. Okay, single explant is nothing but a portion of a plant tissue, whether it can be from root, stem or leaf. So from a single explant, several thousand plants can be multiplied, that too in a very short period of time and space under controlled condition. And this tissue culture techniques are used for commercial production of plants as well as for plant research. And plant tissue culture serves as indispensable tool for regeneration. So how to produce the transgenic plants. And apart from this, there are main applications of plant tissue culture. Okay, so the production of improved varieties, conservation of endangered plants, everything can be made possible with this plant tissue culture. So virus-free plants can be produced, secondary metabolites can be produced, etc. So in this chapter, we will learn in detail about the plant tissue culture and its awareness on ethical issues. In this part of the video, we will learn in detail about the plant tissue culture and its applications. So Gottlieb Haberland, 1902, the German botanist. Okay, so he proposed the concept totipotency, and he was the first person to culture the plant cells in artificial condition. So using the mesophyll cells of Lamium purpureum. So mesophyll cell is nothing but, so in the leaf, the tissues that are found in between the upper and lower epidermis is called mesophyll tissue. And obtain cell proliferation, and he is regarded as father of tissue culture. Now let's see what are the basic concepts of tissue culture. So the basic concepts of tissue culture are totipotency, differentiation, dedifferentiation and redifferentiation. The first concept is totipotency. So what is totipotency? The property of the live plant cells that they have the genetic potential. That is, the plant cells have a potential, that is, if it is cultured in a nutrient medium itself, it can give rise to a complete individual plant. So here you could see a portion removed from this plant carrot. Okay, this serves as an explant. The explant is cultured in nutrient medium. It will lead to the production of callus, unorganized mass of cells, callus, and which can be induced to give rise to a complete new plantlet. Okay, so this property, okay, it is a very unique property of plant cells. No other living organisms can be cultured like plant cells. So animal cells and all, it cannot be cultured. Of course, this is the very unique property of plant, totipotency. The next step, concept of tissue culture is differentiation. So differentiation is nothing but the cells will become specialized, okay, and take up its form and function. So it is called as differentiation. The third concept is redifferentiation. So the differentiation of already differentiated cells into another type of cell. For example, the component cells of callus have the ability to form a whole plant. Okay, so the callus can give rise to a whole plant in a nutrient medium. This is called redifferentiation. The next concept is dedifferentiation. that is the reversion of mature cells to meristematic state. Okay, so this will lead to the formation of callus. And these two phenomena of redifferentiation and dedifferentiation are the inherent capacities of living plant cells or tissues. And these concepts are referred to as totipotency. So the reversion of mature to again meristematic state. So these are the basic concepts in tissue culture. Next we shall move on to plant tissue culture. So plant tissue culture is used to describe in vitro and aseptic growth, that is apart from any avoiding any contaminants. Okay, absence of sepsis. Sepsis means contamination of any microorganism. Absence of sepsis is called aseptic growth. So to grow the plant part on tissue culture medium, and this has several fundamental features. First, the explant has to be isolated from the plant. The explant should be maintained in controlled environmental condition, that is physical and chemically defined nutrient condition. And aseptic condition should be maintained. Okay, so because if it is not maintained, it will lead to contamination of the plant cells. So aseptic condition should be maintained. What are the laboratory facilities for plant tissue culture? So for plant tissue culture, the laboratory should have the following facilities. So first, washing facility should be there for glassware, and ovens should be there for drying the glassware. After washing, it has to be dried. The medium preparation room. So if the medium preparation room should have the following. One is autoclave, an apparatus for sterilization, electronic balance for weighing the nutrients, and pH meter to check the pH, and accordingly we can adjust the pH. And we have to have a sterile room, laminar airflow bench. So this is the laminar airflow bench. So it should have a filter, HEPA filter, high efficiency particulate air filter. So under this condition, we can maintain the aseptic environment, okay, for the process to be done. Next, culture facility. The plant should be inoculated into culture tubes at 22 to 28 degrees Celsius. Growing the explant, inoculated, okay, and it should be incubated properly at a proper temperature and with a proper photo period, okay, and illumination. So photo period of 8 to 16 hours should be provided, and a humidity of 60% should be maintained. These are the laboratory facilities for plant tissue culture. Now let's move about the techniques involved in plant tissue culture. First is sterilization. Sterilization is the technique employed to get rid of microbe. Okay, so the microbe should be get rid from culture medium, the vessels and the explants. Maintenance of aseptic environment. During in vitro tissue culture, maintenance of aseptic environmental conditions should be followed. First, we have to sterilize all the glasswares that we are going to use, forceps, scalpels and all accessories, and in autoclave. So this is the autoclave. So this works on the principles of wet steam sterilization. So it works under at high pressure and high temperature. The microorganisms will be killed. So that is the basic principle behind this apparatus called autoclave. So the pressure is 15 psi, and at this pressure the temperature inside can be raised to 121 degrees Celsius, and when it is kept for 15 to 30 minutes, so completely it will be made free, okay, of all the contaminants, if any. Or how can also you sterilize? Another method for sterilizing like forceps and all, we can dip in 70% ethanol, and then it should be flamed, and then it should be cooled. Next, how to sterilize the culture room. Okay, the floors and walls are washed with detergent and then with 2% sodium hypochlorite or 95% ethanol. And the cabinet of the laminar airflow, okay, sterilized with 95% ethanol, and UV exposure will also kill the microorganisms. Sterilization of nutrient medium. The culture medium which is dispensed in glass containers, it should be plugged with non-absorbent cotton. So this is called as plugging. Okay, so you can see the conical flask is plugged with cotton. So plugged with non-absorbent cotton, because if we plug with absorbent cotton, it will all the water can be absorbed during the wet steam sterilization and leading to contamination. So we have to plug with non-absorbent cotton or sealed with the plastic closures, and it should be sterilized at this 15 psi pressure, 121 degrees Celsius for 15 to 30 minutes. So this is for nutrient medium. How the extracts, vitamins, amino acids, hormones are sterilized? Because when we are sterilizing it under this temperature, all the activities of these will be lost. So to avoid that, we are going to sterilize the vitamins, amino acids, hormones, etc. by using a filter. That is the millipore filter, and it have 0.2 micrometer, the pore size. Okay, and when we filter these substances, if any microorganism is there, it will be stopped by this filter. Okay, and then when we are adding the vitamins or amino acids or hormones to the culture medium, it should be done under sterile condition. Next, sterilization of explants. The plant material to be used for tissue culture should be surface sterilized. That is, first exposing the material in running tap water and then treating in surface sterilizing agents. So that is, we cannot sterilize the explant by any of the above method. That is, we cannot use ovens or we cannot use autoclave. So it can be only surface sterilized by using chemical substances like 0.1% mercury chloride, 70% ethanol, and this has to be done under aseptic condition inside the laminar airflow chamber. So what is the explant? The tissue taken from the selected plant transferred to a culture medium and which is used to establish a new plant is called explant. Next is media preparation. So the success of the tissue culture, it depends on the medium, the plant regulators that are added, and the culture conditions that we are going to maintain. So with a single medium, we cannot achieve the optimum growth of plant tissues. So suitable nutrient medium should be produced and it should be used. The most common plant medium used for plant tissue culture is MS nutrient medium. Murashiji and Skoog. In 1962, they devised a medium. And it has carbon sources, suitable vitamins, hormones, etc. Apart from MS medium, what are the other mediums used for plant tissue culture? They are B5 medium, white medium, NITS medium. And the medium that is used, it can be either a solid, semi-solid or liquid. So for converting a liquid medium into a solid medium, that is solidification, a gelling agent can be added. The gelling agent is agar. So what is agar? Agar is a polysaccharide. So it is a mucilaginous polysaccharide. It is obtained from marine algae, okay, seaweed, Gracilaria, Gelidium. So from that it can be extracted, and this can be used as a solidifying agent in media preparation. The next is culture condition. So after the medium is made, the pH should be adjusted to 5.6 to 6. And the temperature in the incubator, what is the temperature that we are to maintain is 25 degrees Celsius plus or minus 2 degree for optimal growth. Humidity, okay, 50 to 60% of humidity, and 16 hours of photo period, and illumination can be given by white fluorescent tubes of 1000 lux. Next, aeration. Okay, so when the aeration can be provided by shaking the flask or the tubes. When it is liquid medium, it can be the tubes can be shaked, okay, or air can be passed. So for shaking, this is the apparatus. It is referred to as automatic shaker. So it can be kept, and if it is switched on, okay, it will produce agitation, and the agitation that is produced will make the medium aerated. Next is induction of callus. So after the explant of 1 to 2 centimeter, the sterile segment, whether it is a leaf, stem or a tuber or root, after it is inoculated in the nutrient medium, the medium is also supplemented with auxins and properly incubated in proper light and photo period, it will lead to the formation of callus. So callus is nothing but mass of unorganized growth of plant cells or tissues in vitro culture medium. So this is called as callus. Next is embryogenesis. So from the callus, somatic embryos known as embryoids are formed, and the embryoids are subcultured to plantlets. So this process is called as embryogenesis. Next is hardening. So after the plantlets is produced in vitro, it requires hardening period. That is, it has to be gradually exposed and it should be transferred to greenhouse or hardening chamber and then to normal environmental condition. And hence it can be called gradual exposure of in vitro developed plantlets. That is, the plantlets that are developed in laboratory, in humid chamber, in diffuse light for acclimatization, because it has to adjust itself to the normal field condition. So this process is called as hardening. So this is the basic steps in plant tissue culture. This can be asked as a diagram. So from the plant, an explant is taken and it is added in the nutrient medium, and callus is formed, and from the callus it is separated and single cell cultured, and from the single cell we can produce the entire new plantlet. So this is the steps involved in plant tissue culture. So in this part of the video, we have seen in detail about the steps involved in plant tissue culture. Meet you in the next video. Thank you.